really appreciate to talk about fungi and fungal resistance. And often I think it's not as well known as bacterial resistance, so I'm happy to share some data with you. These are my disclosures. So to start off, fungi are everywhere. So if we would have some of these agar plates and we put them on the floor here, and you culture them, even if you vacuum clean it, you will find a lot of molds. So, and the problem is that some patients, like this patient, is susceptible to infection. And there are a lot of fungal species, estimated is 6 million. But fortunately only 600, which is still quite a lot, cause disease in humans. And one of the reasons is that a lot of fungi can't grow at body temperature. So 37 degrees is too high for most fungi. So when we talk about fungi, you usually have two differences. One is yeast and the other is mold. And yeast often we find in association with animals or humans. And molds are usually, they come from the environment. So they grow in the environment and then patients or people are exposed to their spores. So if you think about resistance in fungi, this is much less known than in bacteria, but there are 4 fungi, I think, which have a problem with resistance. Yeast, Candida albicans, Aspergillus fumigatus and mold, and other yeast, Candida auris, and also dermatophyte, which cause superficial infection. So some of these are due to human exposure. So Candida glabrata, it can become resistant if a patient is treated. But others like Aspergillus fumigatus and probably also Candida auris, it's due to changes in the environment. So in the Netherlands, I think Aspergillus fumigatus is one of the main concerns, and I will focus on that today. So this is the fungus, Aspergillus fumigatus, which grows on an agar plate, but in the environment it grows on decaying plant material. So it doesn't actually attack living material, but decaying plant material. And when it grows, it can make these structures which have spores, and these spores are released into the air, and we all inhale these spores during the day. So we don't get ill from those spores, but some people do, and it depends on your immune system. So if you have immune dysfunction, then you get the most severe infection, and we call that invasive aspergillosis. So it grows invasive into the tissue. In patients who have chronic lung disease or have had tuberculosis, you could have an aspergilloma or chronic forms, but you also have people who have hyperreactivity to spores, and they have an allergic reaction, and then you have some allergic syndromes. So in invasive aspergillosis, you could have a lung like this. This is a CT scan, and you can see here the lung fields, which should be black, but you can see that there are white points there, and also here it's like a cavity. And if you would look in the tissue, you can see that here this is the fungus growing, and around that you have bleeding. And if you look under a microscope, you can see the hyphae of the fungus. So that is how it causes invasive disease in patients. So we know quite a lot about this fungus, and we know that patients, for instance, with leukemia, they have a risk to develop invasive aspergillosis, but we also see changes over time, and this is one patient who was admitted to our hospital with very severe lung damage due to aspergillus. And it turned out that this patient had influenza. So this is aspergillosis in association with influenza, and this is something we only recognized only a few years ago. So this is a very unfavorable combination, influenza and aspergillus. Why is that? Well, if you look at the patients we found when we looked in intensive care units in the Netherlands, we found that if you are admitted to the ICU with influenza, 1 in 5 actually developed invasive aspergillosis, which is really high. We were surprised by that. Also, we found that 1 in 3 patients had been healthy. So no leukemia or other underlying conditions, they had been healthy. resistance was an issue in 1 in 3 patients, and also 1 in 2 died. So this is a really severe infection. So one of the problems with fungi, which is different to bacteria, is that if you look at the fungal cell, it's quite similar to the human cell. So they're eukaryotic cells. So that's the problem, is that you find a target to try to kill the fungus, you probably have a similar target in the human cell, and that is then toxicity of the drug. So that means we only have very few drugs we can use to treat patients. So we have the polyenes, the azoles, and echinocandins, and these are not very good for Aspergillus treatment. So we have voriconazole, which is an azole, which was licensed in 2002. Isavuconazole, licensed in 2015. And the alternative treatment is amphotericin B, which is an older drug, but that is considered a little bit less effective than the azoles. And we also have an azole, posaconazole, for prophylaxis. So what is the impact of resistance? Well, this is a patient who has a leukemia, acute lymphoblastic leukemia. And the survival probability of this patient after 1 year of treatment of this leukemia is 94%. She is at risk to develop invasive aspergillosis, and if it's confined to the lung, the survival is 70%. If it spreads to the brain, then you have a much more serious infection, but it's still 50%. But if it's a resistant isolate, then the survival chance is 0. So there are patients now who develop this disease who have actually no treatment options. Why is that? Well, the first problem is that it's quite difficult actually to find resistance really early. So we don't have good tests to do that. And the other problem is that we don't have an alternative drug which actually gets into the brain well enough to actually treat this infection when it's in the brain. So in general, if you look at the clinical implications of resistance in Aspergillus, we performed a cohort study in the Netherlands in 3 medical centers, Radboud, Leiden, and in Rotterdam. And we screened all the patients to see if their cultures were resistant or not, and then we looked at the survival of those patients. So we had 196 patients, and 37 of those had a resistant infection, and the others had a susceptible infection. Most of these were treated with voriconazole, which is our first-line treatment, nearly 80%. So when we looked at the survival, which is done at 6 weeks, we found that that was significantly lower if you had a resistant infection, which is 20% lower. So if you look at the efficacy of the azoles, and this is voriconazole, you can see that in most studies which have been done since 2002, you can see that the survival rate is between 60 and 70%. And when we look at our study and another study, you can see that the patients with a susceptible infection actually were exactly within this range. But if you have a resistant infection, actually the survival is much lower, and even lower than before we had the azoles. So the benefit which we had from the azoles is actually totally lost due to this resistance. So this gives us problems because we can't use voriconazole in these patients, we can't use isavuconazole, and we can't use posaconazole. So we lose the class. And that gives problems because we don't have drugs left which actually penetrate into the brain very well. And the other problem is that we have no oral drugs. So patients sometimes have to be treated for months, and we don't have any oral drugs, so they need to have an intravenous drug, and that is also very complicated often. So how does resistance emerge? Well, actually we recognize 2 routes, and the first route is the patient route, and the other one is the environmental route. So in patients you can have both routes. Often we see in patients with chronic disease, like aspergilloma, we see these are often treated with azoles, that they get the patient route resistance. But patients with invasive aspergillosis in the lung, associated with influenza and central nervous system, it's always the environmental route. So what is the environmental route? Well, we have found that azoles are not only used to treat patients, but are also used in the environment, for instance, to prevent crops from fungal disease. And some of these are triazoles, and triazoles are also used to treat patients. And aspergillus is present in the environment, and we think that due to exposure to these azole fungicides, you get mutations. And when the patient inhales these resistant isolates, we try to treat them with the medical triazoles. They don't work because you get cross-resistance to the medical triazoles. So that makes actually it much more complicated. And the other thing is that if you look at the lung in patients, we also find that some of these lesions are caused by susceptible aspergillus and some are caused by resistant aspergillus. And that really also complicates management, because if you have a culture, you have to test all different colonies to be sure that there's not a resistant one among the susceptible ones. And also if you treat these patients, which we have done, with an azole, they actually improve, but you only treat these lesions and then these lesions progress and they disseminate. So that is a problem for the management of these patients. So can we diagnose this in these patients? This was the study we did. We had 154 patients who were treated with voriconazole. That was our first line treatment. We have then a positive culture, and then we look with an MIC test if it's susceptible or if it's resistant. And if it's susceptible, then you are treating appropriately, but if it's resistant, then you have to switch to other treatment. So this was in the setting of intensive screening of these isolates, and we found that it took 10 days actually to make this switch. So first you need a positive culture, then you do an MIC test, and that takes a lot of time. And we also found that the survival in patients who had the wrong treatment first, it was actually 20% higher. So the initial treatment has to be right, otherwise you will lose patients because of this delayed treatment.
And Paul, on the exit was the time?
Yes, so this is the time after start treatment, and this is the survival. So what we have done in the Netherlands, we decided to change the guideline in 2017. So we say it's really important to diagnose the infection, to know that it's actually Aspergillus fumigatus, to do a PCR or do an MIC testing. And then we have 4 possibilities. It might be susceptible, it might be resistant, it might be mixed, or we don't know. And we only recommend now the azoles if it's confirmed susceptible. If it's resistant, we confirm the other drug, and for the other options, we actually recommend combination therapy. So this has major implications. We hope that we treat patients directly with the correct drug, but at the same time we recognize that we really overuse the drug because 85% of these patients will have a susceptible infection. So this has made management of fungal diseases much more difficult, and in the Radboudumc we have a center of expertise in mycology where every 2 weeks we have a multidisciplinary meeting. We have specialists, infectious diseases in pharmacy, mycology, pulmonology and hematology. So we just discuss patients and also patients from outside Nijmegen through video conference, and we discuss how to manage these patients. So we do surveillance in the Netherlands, and this is 5 university medical centers where we screen isolates cultured from patients just to see what the trends are. And you can see that in these centers, this is Nijmegen, it's nearly 12%, Rotterdam 13%, Leiden is also always high, 20%, Amsterdam 16%, and Groningen is 14%. So this is in university medical centers, and also recently we included non-university medical centers, and maybe Herjan can comment on what they find, but it's lower there than in university medical centers. Did you have a question?
Yeah, it was resistant, but it's in the title.
Yeah. So, and also important is that most mutations are actually from the environment, 80 to 90%. So that's really an important route for resistance. So it's not only the Netherlands, also in other countries in Europe, resistance is found in India, China, Australia, and also North and South America. But you can see that a lot of countries are white, and in white countries we have no idea about resistance. So, is this part of AMR? Well, if you look at AMR research, and this is a leaflet from the United Kingdom, it says AMR, but if you read what it says, it talks about bacterial resistance. And if you look at bacterial resistance, for instance, in the Netherlands, and this is with resistance in pneumococci, you can see that we are doing a really good job. But the problem is that AMR, it's not ABR, it's not antibacterial resistance, because it's antimicrobial resistance. And that is a problem because I think there's not enough awareness of the problem in fungi. So I just did an analysis for the Dutch Medical Research Council in their infectious disease programs in the last 14 years. And you can see that in this program, fungal resistance were tolerated, but here they were excluded. And in the recent one, it was also excluded, and this one also excluded. So overall, 129 projects in infectious diseases were awarded, but only one in fungi. So I think it's really underestimated. Why is that? Well, the first reason is that fungal diseases are not recognized as a public health disease. Bacteria are, but for fungi, you need an additional factor, like some problem with your immune system. The other problem is that if you look in agriculture, Aspergillus fumigatus is not a plant pathogen. So fungicides are not directed to Aspergillus. It's not a target, but it's actually in the waste where it causes a problem. And also, MIC testing in medical microbiology laboratories is not done routinely. Unlike bacteria, where it's done routinely, for moulds it's not done. So that is, I think, some of the reasons that there's a lack of awareness. So, but there is one CDC in the United States where they do have interest in mycology, and they recently, in November, they published a list of urgent threats in antibiotic resistance. And for the first time, there was a mold on this list, Aspergillus fumigatus, which is on the watch list. Candida drug resistant is a serious threat, and Candida auris an urgent threat. So it is emerging there as a problem. So I want to spend my last 5 minutes on the environment, what is happening in the environment, and sometimes it comes in the news. Deadly flower power, important tulip bulb spread antifungal resistance. So what is happening in the environment? Well, together with Wageningen University, we did some research in the environment, and we defined what we call a hotspot. And a hotspot is a condition where the fungus can grow and make spores, but also where azole residues are present. And we investigated a number of areas, and we found a few hotspots. So the flower bulb waste is a hotspot, green waste is a hotspot, wood chippings is a hotspot, and recently in China they also found that strawberry waste is a hotspot where you have not resistance. So if you measure in these environments and look for azoles, we found that a lot of these contain fungicide residues, and a lot of different ones. Some had up to 8 different fungicides. And if you look, there are a lot of different ones present, but most frequently we find these triazoles, which cause cross-resistance to the medical azoles. So the medium concentration was low, but it could be very high in some samples. And also we found that Aspergillus really likes to grow in this compost. So the next question is what happens then? Where does it happen? And to know that, you have to look at the cycle. So fungicides are applied for plant protection. What the farmer then does is collects his waste and leaves it on the land in a big pile for 8 or 10 months. And then they do active composting, and then you have mature compost, and that is used again to fertilize the ground. So where in this cycle is the problem? Well, we looked at commercial composting, where grass and leaves and wood chippings, they are mixed. Then they are processed, where the temperature rises, so they mix it around, and you end up with mature compost. So if you look at Aspergillus, this is susceptible and this is resistant. You can see that already in grass, which is used as input. You have a lot of resistance. Also in wood chippings, but interestingly in leaves there was no Aspergillus. So apparently Aspergillus doesn't like to grow there. But if you look at the processing, actually you see that the resistance go down, and in mature compost we found much lower Aspergillus and no resistance. So composting is good, and that's directed of course against the plant pathogens, because it kind of sterilizes it so that you can use it again to fertilize the soil. So mature compost is not the problem, and we think that the storage that is the problem, and we need to, I think, look at interventions how to change that. But the problem is, you go to this farm field and you have this heap, and how does the patient get infected? And that is something we don't really know yet. Could it be that if you give flowers, maybe you give resistance? I always say, if your mother-in-law is in the hospital and you give tulips, is that a good message or a bad message? But there might be other routes of transmission. For instance, in tea, if you just have tea leaves and you put them on the agar, you get Aspergillus growing, including resistance. Also in pepper, it's been found in coffee, in onions, in carrots. And then you come much closer to the human. So this might also be an important route of transmission. Water, of course, is another question which is interesting. That is what we don't know. I've had some collaboration with the Water Institute, the KWR, and they had 180 fumigators isolates from water samples, and we found that 5% were azole resistant. And the other question is, of course, is there exposure? So is there a selection pressure in water? They told me that all these azoles are taken out of the drinking water, but that is an area that I don't know, but that could be another concern. So in conclusion, I think that this is a threat. We really need to have better diagnostic tests in the management of patient diseases, and also new drug targets. It's a global problem, so unlike bacteria, there are no international surveillance programs, and I think that's important. And also One Health programs are important. And also I think it's important to include fungi and fungal resistance in AMR programs, and also to prioritize that.